to talk about one of the roles I had throughout the pandemic. I worked very closely with Thomas and Ian, but they were already busy with other things, so I was the only one who could pick up this task. And at the beginning it was fun, but then it stopped being fun. And so I was responsible for submitting the reproduction number estimates, you know, the R number that everyone has been quoting. There have been multiple teams submitting that to SPI-M, and then things were merged together in some sort of model combination to produce something that was then published by the government. So how it all started, I think in 24th of March 2020 was the first lockdown. At the end of March, I remember, I mean, clinicians were probably busy in other ways that I find it hard to imagine how busy they were. But our eyes were very much glued on how hospitalizations were growing, and we were really hoping the hospital capacity wouldn't be breached in London. In the start of April, modelers explained the concept of this reproduction number to some of the politicians, and just in case, I mean, you probably, I'm sure you know about that, but this is so the reproduction number is the average number of secondary cases generated by a single case during their entire infectious period. Okay, and then we can distinguish between different types of reproduction number, whether the population is fully susceptible or not. But then the politicians were like, "Oh, this is amazing. We absolutely need to publish our best estimates of this number." And so there was an immediate call to army, and I remember, like, I coded up. I didn't have a model ready, so I coded up the model between 11 p.m. and 5 a.m. Together on a call with someone in Canada who was just checking I wasn't making any mistake, and I started submitting estimates from the next day. The process became very quickly very formal. So the data was coming in on a Monday. We were all fitting our models in many different groups on a Tuesday, submitting our model estimates by Tuesday night. Then there was a meeting on Wednesday to discuss the results, fixing something if needed. Then on Thursday, it was kind of approved by SAGE, and then on Friday, it was published by the government. And that cycle repeated, okay. And then we got stuck in this task. It became such an official thing that it was kind of quite difficult to get out of it. And it was a very routine thing. So UK HSA then took it on board, making it a routine thing rather than an academic job. And but the handover was very slow. And then even in UK HSA, stopping this process was very difficult. You had to have some people saying that we just should, after 3 years, we should just stop doing this because no one needs it anymore. Ian already mentioned something about the pandemic response. It was SPI-M, SAGE, the Cabinet Office briefing room, but there were also a couple of dedicated subgroups, not really formally, but there was this medium-term projection group, and then which then became the UK HSA epidemiology modeling review group. And we were submitting both estimates of R and the growth rate and this medium-term projection. So we were just trying to project forward how many hospital admissions and beds occupied in hospital or deaths we were seeing with the models. It was a very fast response, moving target situation. There was a lot of team effort because everyone could get stuff wrong every now and then. So there was this model combination that was trying to put together the output of all the models in order to kind of minimize our reliance on a single group. And so this is a picture of what could have, it's a very blurred one, of what kind of was happening. Different models had different levels of accuracy and different estimates. And then the black one is kind of some sort of model averaging of all of these outputs here, okay? I'm not gonna go into that process because I wasn't responsible for that, and lots of clever people were doing that part. So this is an example of what was happening. We were meeting, we would have some information about the data. This was the data that was submitted to us. We were all trying to fit the model for the different countries or the different regions in England. And as you can see, different models were picking different sort of signals in the data. Some people were just interpreting all this data as kind of flat and then continuing the trend. Some other people were just picking this as an uptick and were kind of going up and predicting a growth. And then we were kind of combining all of these models. Then when there was a time when there was a major change in control policy, like a lockdown or coming out of a lockdown, like in this particular example, then we would see, we knew that the transmission was changed, but we wouldn't have seen that signal in the data yet. So we were kind of making some assumptions about what the transmission might be and try different values of the reproduction number going on in the future. And then we were trying to kind of explore what the potential futures were. We were calling these scenarios. This is just to give you a kind of a illustrative picture of what's going on. The model we used in Manchester was this so-called EpiBED model. I'm not going to spend much time over it, but I wanted to say a little bit about the modeling so that people are more or less interested in that can kind of ask some questions later on. So we were using the CHESS, now called the SARI dataset. These are incomplete data set of a subset of hospitalized cases, but they have quite a lot of information about hospital admission date, ICU, leaving hospital, and so on. And then we were using that data to estimate some sort of rates of moving between hospital compartments. So entering in hospital and then progressing into ICU, how long does that take? Moving leaving ICU and stepping down to a monitoring phase, how long does that take? And then we were trying to fit the resulting model that I'll talk about in a second to the NHS situation report data, which is rather complete data about number of hospital admissions, and number of beds occupied and ICU beds occupied. I'm going to skip lots of the details. These are the rates that we were estimating. This is not a trivial task, so it required a bit of thinking. And then this is kind of the model structure. This is sounds really complicated, but in fact it's a very, very, it's a glorified SIR model, susceptible, infected, recovered model, which is a standard epidemiological model. Susceptible people would either be asymptomatic and follow this route, or they would be symptomatic, have a bit of an incubation period, then a prodromal period, and then a late part of the infection, then recovering, or they would have this late part of the infection but then end up in hospital. They could go to hospital and then ultimately die, go to hospital and end up in critical care, and then from critical care die, or either step down on a monitoring stage and then recover, or end up in hospital and then recover. And the rates were from the CHESS SARI dataset, and then fitting to the NHS situation reports would give us a value for the transmission rate and some probabilities of going this route or that route. Yeah. So this is basically an SCIR model with a very detailed description of transition through hospital compartments, because this is all the data we had. And all the stuff outside was really not pinned down by data, but I was using reasonable parameters from the literature. But the one that we were really trying to estimate was the transmission rate, because then the reproduction number depends on that transmission rate. We were fitting into first wave and second wave, and going quite fast here. We estimated some sort of reduction, a 25% reduction in probability of dying outside of ICU between the first and the second wave, and a 15% reduction of dying in ICU between the first and second wave. And then we were trying to do, this is an example of if you do predictions for 2 weeks using the data up to the time when you get, when you start your prediction, that's what you would expect in the next 2 weeks. And as you can see here, you will get things wrong because the data is going down and the model cannot do anything else but predicting. The situation keeps going down, but we know we're coming out of a lockdown, so that's one of the points where you would use scenarios to kind of try to second guess what might happen next. Limitations of this model is that there is no age structure, which is quite strange given we all know age was so important. But lots of other models had age structure, and as soon as you add age, then suddenly you have a whole lot of parameters about how the different age classes mix that you have no hope of really estimating properly from the data. So people were doing very sensible things with these models, but I thought if everyone has a model with age, let me just try to build a model without age, so it's easier to handle, and then I'll see how it compares with other models, and it actually performed incredibly well, to be honest. So the other things I was fitting only to hospital data, so I wouldn't be able to really constrain the epidemic outside of the hospitals very well. And then hospitalizations are not infections, so they are a delayed signal, and I only pick up the signal from the more severe cases that are the ones that end up in hospital. But in terms of estimating the reproduction number, that's not a problem, because if there is a constant probability for people to end up in hospital and the age distribution of cases doesn't really change, then if the hospital data is growing exponentially, then it means that the cases are growing exponentially at the same rate. And in reality, we were also hoping to fit to hospital data and overall deaths, but the only way to actually put both of them in the same model would have been to include care homes in the model, because a third of the deaths were just happening in care homes, roughly, and care homes wouldn't feed into the hospital data because people were dying in care homes. So we would have a model that explicitly take into account care homes, otherwise you couldn't fit both death and hospital data in the same structure. You could fit them separately, which is what many other groups were doing, but you couldn't put them together. So we were trying to extend it to care homes, but we didn't really manage throughout the pandemic. We were just too overwhelmed for that. The strength of this approach is that we were, and I've already run out of time, that it was fast and conceptually simple, so we could fit in relatively quickly, and there were no unidentifiability issues. So all the parameters could really be estimated in this model. So it was maximally informed by data. The other thing that I consider very positive is that bad fits were informative about the changes to the underlying system behavior. So for example, if our model was not fitting the data very well, it's because something was changing, like for example, the age distribution of cases. Or more older people were ending up in hospital than you would have expected or you would have seen up to now. And so for me, the take-home message was use a simple and transparent model, because at least you understand what's going on. Lots of other models were so complex that when there was a problem of discrepancy between the different data streams, all of these could be easily fit by just adjusting some other parameters in the model. And instead, in my case, I just wouldn't be able to fit both data streams at the same time. And that was a strong signal that something was happening. Either the data was wrong or I mean, there was some problem in the data, or something else was changing in the system. So I was learning more when the model wasn't fitting the data than when it was. So for me, that was important. There were lots of practical challenges. I've only mentioned one, that the different devolved administrations were publishing very different data, especially at the beginning. So this was England. I could fit hospitalizations, ICU admissions, and then hospital beds and ICU beds occupied. But then I think this was another devolved administration. I'm not going to name them. But at the very beginning, they had cases up to a certain, like hospital admissions up to a certain point, and then ICU beds occupied for a longer time. And you were like, how do I fit? Every devolved administration was a different challenge. And so I'll skip most of this. I left it there in case we need to talk about it. And then just present a couple of considerations. So strength of the reproduction number. Hopefully some of the things I say will maybe ring a bell with you or make you think, oh, I've also thought that. So it's an easy parameter to read because you just basically try to understand if it's greater than 1, you can have, the cases can grow, and if it's not, then they can't. It also, the absolute value means something, because if we can prevent 1 minus 1 over r of transmission, then we can control the spread. So if the reproduction number is 3, and we manage to cut down 2/3 of transmission, then we reduce the reproduction number from 3 to 1, and that prevents cases from growing. But it's a concept that is independent of time. I cannot tell how quickly one needs to act. If I know that throughout my entire infectious period, I infect 3 people, I'm not telling how long it takes for me to infect them. So there is this time independence issue. Obviously, I know that then intuitively I'm infectious for a week or something, and then I know the speed, but I need to have this extra information about speed. Conversely, the instantaneous growth rate provides some temporal trends, so it tells me how quickly I need to act, but it's less easy to read, or if it's declining, they are halving every week. But then when you reach a peak and come down, then the doubling time goes to infinity suddenly, and then it comes down. So it's still a little bit hard to follow. But then both these parameters cannot tell us how much transmission needs stopping. So that's an information that comes from the reproduction number. So both of these numbers have pros and cons. Ultimately, however, in a nutshell, what you do to estimate the reproduction number with a model is you're trying to fit your exponential growth, or at least temporarily you pretend that things are either growing or declining exponentially. And then you're combining that with information about the generation time distribution, so how long it takes for a person to infect someone else. And that's kind of represented by that curve.
So on average, say, I don't know, 5 days, but roughly variable between, I don't know, 1 and 10 or something like this, a curve like that. And so this is temporal information, and then I'm combining with another piece of temporal information to get a parameter that does not depend on time anymore. Okay, so this is what's happening. But the key point is that I still need my data. If my data is going up, I can estimate my growth rate is positive, and I'm estimating a reproduction number that is greater than 1. And if my data is going down, I'm estimating a reproduction number that is less than 1. But basically, I need to see the data going up or going down before I can estimate my reproduction number. And that creates all sorts of confusion because people were complaining, even like very numerous people that I come across, were complaining that the cases were coming down and our estimates of the reproduction number were still greater than 1. And if we're fitting to hospitalization data that is slightly delayed, cases have started going down or hospitalization have not, then people were like complaining the cases were coming down but the reproduction number was still greater than 1. And the answer is, yeah, we need to see the cases coming down first, and then we can estimate a reproduction number less than 1. But if you're reading the reproduction number as to say, oh, it's less than 1, then cases are coming down, you're switching things around, okay? So some positive considerations. R was at the forefront, which was kind of exciting for a modeler. There was great response from modelers, good knowledge transfer, and a lot more thinking and science. And the short-term projections were quite useful. But there was this enormous machinery that in my mind meant we really overdid it, okay? There was a lots of time, person, hours of work invested on this. And I thought the NHS was possibly the best, the most important stakeholder that was using this information, but I'm not quite sure because I asked someone else and then they said that their partner works in the NHS and the NHS had their own internal model. So I was like, I don't know why we're doing all of this sometimes. So it's pointless to use R to say that the epidemic is going up or down because you have to see it going up or down in order to estimate R greater or smaller than 1. The medium-term projections could be helpful to understand how much is going up or down, that's fine, but eyeballing the data really goes already a long way without really having to fit a model. So I sometimes was half joking that my niece could just draw the dots with a pencil and continue the trend, right? So what you couldn't do with a model really was having an expert stamp on this, which obviously is important, as Ian was saying before. And then that could help identifying whether something surprising is happening or unexpected, which obviously is something that I thought was helpful with our model. And then obviously the model also contains this depletion of susceptibles. So if the data is going up, but then lots of people are already immune in the population, the model would turn around. And instead my niece with a pencil will keep going up, right? So that is a big difference. But modeling this is very hard because knowing how much immunity there is in the population is very difficult to capture. So the models try, but different models were making different assumptions, we're capturing this to various degrees. And I regret not spending more time understanding why the model predictions differed. Not talking to each other was a way of maintaining the models as independent as possible, which is perfect for model combination. But why the models differed was something I really wanted to spend more time on, and there was no time or energy to do that. So that's, I spent way too long looking at exponential growth since 2020, and thank you all.